So thank you for that very nice introduction. And just a very small correction. Actually, I moved its mark from Genentech to Rockefeller University. So actually, it's not. There's a small difference. So good morning, everyone. It's my great honor today to join this symposium at the same time to share with you some of recent research work on the pathological axon degeneration. And as everyone here knows, as a unique structure of neurons, axons form this unique structure to transduct action potentials. And this structure is very essential for the transduction of neuronal signals, and actually is a building block to form all the neural circuits around the human bodies. And the proper function of this transduction of action potentials through axons is very important for the neuronal physiological events such as sensing, motility, cognition, and memories. But what is sometimes ignored or kind of overseen in this picture is that some of the axons are very long compared to the neuronal cell bodies, and therefore it poses a great challenge to maintain structure and functional integrity of axons. For instance, the axons that contain or control the movement of our foot actually have their neuronal cell body located in this lumbar part of the spinal cord. And therefore, the axon projection for the lumbar part of spinal cord will need to be going as long as over 1 meter and to reach the tip of their foot. And therefore, it's not surprising that in many neurodegenerative diseases, the axon actually got damaged and lost in many disease conditions, such as Alzheimer's disease, Parkinson's disease, traumatic injuries, Lou Gehrig's disease, multiple sclerosis, glaucoma, and something like that. And in all those diseases, the damage of the axons or the degeneration of the axon structures actually inevitably will cause the blockage of the transduction of action potentials or neural signals down the axons, and will direct cause to the onset of symptoms such as paralysis, dementia, or even the death of the human patients. For instance, here is shown the picture of the spinal cord of the human patient that's tragically died of the multiple sclerosis. As you can see, in the staining of the axon structure by the silver staining of the black spot, you can see that is along the spinal cord structures, there's a large white spot that shows the structure of axon structures damaged through the disease conditions. As you could imagine, such damage of the axon structure along spinal cord structures will cause a severe defect or the paralysis of the human patients. And here is shown another example of the traumatic injury damage, traumatic damage of the sciatic nerve in human patients. Here is shown that control structure of the sciatic nerve without any injury, and you can see there's very tightly packed intact axons or myelin axons in this picture. But after traumatic injury of the sciatic nerve, after a few days or a couple of days, all the axon structure will undergo a very stereotype degeneration, and all the structure are damaged or disappeared in the distal portion of the sciatic nerve. And those damage of the sciatic nerve structures will cause the paralysis of all the legs and will cause a great pain to the patients. And in the past few years, in my postdoc as well as in my current research at Peking University, we have been particularly interested in the signaling mechanism that control the pathological axon degeneration. And the reason why we got so interested in this question is because accumulating evidence has suggested that pathological axon degeneration may represent a yet-to-be-characterized novel death program. And the key evidence supporting this notion is that, first, all research work as well as other work in the field suggests that this axonal death is, in most of the case, is non-apoptotic, means that it's not controlled by the classical apoptotic pathways. Some of the key evidence is that in all the degenerative axons in many conditions, there's no detectable activation of caspases, which is the key executioner of the apoptotic pathways in those damaged axons. And more importantly, through the very comprehensive genetic analysis in your hand, we found that the genetic depletion of the key apoptotic components, such as BAC, caspase 9, caspase 3, caspase 7, caspase 6, do not afford any protections of the damaged axons in many disease conditions. Therefore, the axon degeneration most likely is non-apoptotic. At the same time, our research also suggests that this pathological axon degeneration also is non-necroptotic, which is a newly identified cell death program. The key evidence also includes such as the classical necroptosis inhibitors could not delay any of the degeneration in many disease concepts. At the same time, the deletion of the key necroptotic components such as MLKL, RIP3 have no protection at all in several of the disease conditions we tested. Taking all this evidence together, it suggests that the pathological axon degeneration represents a new novel death program that warrants further characterization. During my postdoc research at Mark Levin's lab at Rockefeller University, we through a very high throughput whole genome screening by shRNA, we first identified a key regulator of the pathological axon degeneration, which we call it SARM1, SARM1. As you can see, here is shown the control condition, which in the wild-type mice will do the sciatic nerve injury. After a couple of days, all the axonal structure will degenerate in the distal portion of the sciatic nerve. And at the same time, all the axonal terminal which innervates the hinder limb muscles, for example, the tibial anterior muscles, will disappear, as shown by the green staining of the terminal of axons, while the red staining shows the structure of the acetylcholine receptors on the muscle. In sharp contrast, in the SARM1 knockout, after the same sciatic nerve injuries, all the axons could return their structure integrity as long as here shows 6 days, but the longest one we have done is 3 weeks. Basically, as long as to 3 weeks, the SARM1 knockout can maintain their axonal structure after the physical injuries. Moreover, even for the terminal structure of the axonal projection to control the muscle movement, you can see the axonal structure of the motor neural axons could remain intact as long as to 6 days after the traumatic injuries. This is the first evidence of the loss of function study showing that the pathological axon degeneration is actually controlled by a genetically defined pathway, with the SAR1 being a central regulator of this program. And then, to answer the question as to how the SAR1 transduces the degeneration signal to the downstream event, we continue our screening by the shRNA library, and then we were able to identify a classical MAP kinase pathways composed of the 3 MAP3K kinase, 2 MAP2K kinase, and 3 MAPK kinase that function as the central downstream event or signaling transduction modules downstream of SAR1. And here shows the example of the optic nerve injury. As in the control condition in the first column, you can see that after the traumatic injury of the optic nerve, after a few days, all the axon in the damaged optic nerve was degenerated and completely fragmented. But in the sharp contrast again, when we knock down or knock out together with the JNK1, JNK2, JNK3, all the 3 MAP3 kinase here, all the axons could return intact as long as to 6 days after injuries. This suggests that the SAR1 is likely to function through its MAP kinase pathways that regulate the degenerative process. And then we try to tackle the question as to how this SAR1 MAP kinase pathways could transduce the signal further downstream to cause the fragmentation of axons. To this end, we observe a very interesting phenotype that we're going to elaborate on in further talk, is that it appears that the pathological axon degeneration seems to have close connection with the metabolic status of the axons after the injury. It means that we were able to directly measure the axonal ATP levels after the different time point of the injury. As you can see in these blue lines, after different time point after injury, there's a very dramatic drop of the ATP levels, very quickly drop of the ATP levels in the damaged axons. And it appears by this Western blot correlating with its ATP drop, that when the ATP level reaches the final state, which is basically undetectable level, the axon degenerates completely with the complete cleavage of the main axonal structure proteins. And to support that this ATP depletion is the causative event of the axon degeneration, we play some trick that we're able to return or maintain the ATP level in damaged axons by providing the axons with some metabolic intermediates. For example, here is shown the derivative of methylpyruvate, pyruvate, which is a derivative of the key product of the glycolysis, and it's also a very important fuel to feed into the TCA cycle. As you can see, when we're able to treat the axons with this methylpyruvate, as shown in these red lines, they can it's very significantly delay the drop of ATP levels after the damage. Not only so, this delay of ATP drop correlate with almost a complete protection of the axonal structures after the damage of the axons. This suggests that the ATP depletion is a causative event after the damage of the axons. Moreover, we were able to show that this energy depletion or energy deficit is actually a function downstream of the SAR1 MAP kinase pathways. Because when we do the wild-type control, we can see this ATP depletion happens, as I showed just in the past slides, happen very quickly after the damage. But in contrast, in the SAR1 knockout, or when we knock out or knock down the JNK2, JNK3, or JNK1 together, there's also a very dramatic delay of this ATP depletion. suggesting that very likely this SIR1 MAP kinase pathway function to control this local energy deficit event that caused the downstream event, caused the degeneration of axons. Then, as the final stage of how the connection of ATP depletion is basically how the ATP depletion is connected with the physical fragmentation of axons, we have very convincing evidence suggest, just for the sake of time, I'm not going to show you most of them, is that after the ATP depletion, there's accumulating of calcium inside of axons, which is not surprising because, as I will know here, the memory potential of the axons or neuron as a whole is need consistent input of ATP to be maintained. And with ATP depletion, therefore, this loss of the maintenance of this ion balance across membranes will cause accumulating of calcium inside of axons. And this accumulating of calcium inside of axons will eventually cause activation of a classical calcium active protease called calpains. And this calpain activation will cause the final fragmentation of axons. In support of this idea, we first use a genetic approach to delete the calpain activity through this calpain 4 conditional knockout. As you can see, in these optic nerve injuries models, in the control condition, after a few days of optic nerve injuries, all the axons degenerate and fragmented. In sharp contrast again, with this calpain depletion or inhibition of a calpain signal, there is almost a complete protection of the axonal structure up to even to 6 days after injuries. Moreover, we were able to show that this calpain indeed functions downstream of ATP because, by a very simple trick, we were able to use the calpain inhibitors that inhibit the calpain activities and strongly protect the axons, both in vivo and in vitro. But these calpain inhibitors have no effect at all to delay the ATP depletion inside of axons, as shown in this red line compared to the controlled blue lines. Supporting our hypothesis, this calpain activation is functioning downstream of ATPs. And therefore, in my previous postdoc research in Dr. Marcus Levin's lab, we were able to delinearize a novel signaling pathways starting from the SIR1 to downstream MAP kinase pathways to further downstream ATP depletion event and leading to the final activation of calpains that cause the fragmentation and the degeneration of axons. And when I move on to my next job and my working in Peking University last years, I strive to answer one of the key questions in this diagram, is that how the local energy homeostasis got disrupted in damaged axons. Basically, what is the causal event for this ATP depletion after the damage? And to this end, we were able to, first to refresh your mind, hopefully you still remember about 101, that is, the energy homeostasis in the mammalian cells, I say it's mammalian cells, it's not in plant. In mammalian cells, is mainly supported or is solely supported by the two major pathways that can generate ATP. The first one is called glycolysis. And through the degradation or partial metabolize of the glucose, will generate a portion of ATP. And the end product of the glycolysis pyruvate will then feed into the TCA cycle. And through a TCA cycle and the electron transportation chain, will generate a large amount of ATPs. And because we already know in the damaged axons, there is an ATP depletion or energy deficit, we therefore try to understand which portion of this energy homeostasis got disrupted, whether it's glycolysis or whether it's the TCA cycle. And for the sake of time, I'm going to show some very brief results. And we were able to use a very small trick, but a very effective trick that can analyze the event wholly going on in the axons by surgically remove the neuronal cell bodies through in the in vitro cultures. By doing so, we were able to focus solely biochemically on the event or the energy status in the axons after injuries. And by doing so, we first ask question whether the uptake of glucose for the axon may be affected after the injury. And the answer is no, because we can use a very classical radio-labeled glucose uptake assay through the C14 labeled 2-deoxyglucose. As you can see, after injury of the axons, there's no significant defect of the glucose uptake in the axons. And for sake of time, I'm not going to show you, we were also able to analyze the TCA cycle efficiency in the injured axons. And the answer is still no, means that there's no clear defect of the TCA cycle in the injured axons up to the time point with ATP depletion already occurs. Therefore, we put our focus onto glycolysis. And to do the glycolysis analysis, we use a very classical and very old trick, is that we can reconstitute the glycolysis event in the test tube. To do that, we can isolate the axonal proteins, cytosolic protein of axons, all the cytosol components of the axons. And then we could provide this mixture of cytosolic components of the axons with intermediate glycolysis, which is the fructose 1,6-biphosphate. By doing so, we can reconstitute the production of ATP completely in vitro. As you can see, with the control condition, there's no ATP generation at all, as is shown in the blue line. But when we provide this in vitro reconstitution system with fructose 1,6-biphosphate, there's very rapid and very efficient generation of ATP in vitro. And then, when we use this assay to isolate the axonal structure, the axonal contents, isolate from different time point after the injury, which is a 0 time point or the 4 hours after injury. As you can see in the red line compared to the blue line, the axonal content isolated after the injury, 4 hours after the injuries, shows significant defect in the production of ATP through the glycolysis event. This is the first indication, that is, there seems to be a blockage or impairment of the glycolytic ATP production in the injured axons. But the question is why? And before going to the question why this happens, we first ask the question is whether this glycolysis defect is also downstream the thermal MAP kinase pathways. And to show some of the data here is that we found that this blockage of the glycolytic ATP production is actually depends on the thermal MAP kinase signal. Because, as I just showed in past slides, in the wild-type axons, if we do this glycolysis in vitro assay, the glycolytic ATP production drops very rapidly after injury. But in contrast, when we have the thermal knockout axons going into this assay, they almost completely maintain their ability for this glycolytic ATP production up to even 6 hours after injury, which there's already significant delay, there is significant decrease in the wild-type axons. And then with that, we're trying to explore the reason why there's a blockage or impairment of glycolytic ATP production in the axons. And just to make the story short, we found that actually there's a very central cofactors that is essential for the glycolysis going through from the fructose 1,6-biphosphate to the ATP production, which is the NAD. And this NAD is an essential factor to the step catalyzed by the GABA-DH or the glycerolaldehyde 3-phosphate dehydrogenase. And we use a very classical but very efficient way to measure the ATP level in the axons through the radio labeling of the ATP through the radio labeling of the NAD precursor nicotinamide. As you can see, after injury, when we measure the NAD content in the injured axons, there's also a very rapid drop of the NAD level inside of axons. Most notably is that this NAD drop inside of axons is actually in parallel with ATP drop inside of axons. This is an indication that this NAD drop or NAD depletion happens inside of axons may be related or physically connected with the ATP depletion inside of axons. To test this hypothesis, we play a very simple trick. We can add the exogenous NAD into our in vitro glycolytic assays to see whether this exogenous NAD could rescue the glycolytic ATP production in vitro of the injured axons. As you can see here, the white bar, as I just showed in past 2 slides, after injury, there's a dramatic decrease or significant decrease of the NAD production through glycolysis inside of axons. But if we provide the exogenous level or the source of NAD into this assay, the ATP production through glycolysis reaction could almost be completely rescued. It suggests that the depletion of axonal NAD is actually causative to this blockage of glycolytic ATP production. But then the next question is why the NAD got depleted. And to refresh your mind again of the basic biology of the NAD metabolism, in the mammalian cells, the NAD is mainly synthesized through a recycling pathway. It's through the precursor of nicotinamide. And the nicotinamide will be condensed with ATP first to generate an intermediate called nicotinamide mononucleotide. And the nicotinamide mononucleotide will be further condensed with with ATP to generate the final product of NAD. And this NAD, in certain conditions, will be break down or consumed through certain class of NAD consumption enzymes I'm going to talk about later that will break the NAD back into the precursor nicotinamide. And if that's the case, then we ask the question, why the NAD drop inside of axons after injuries? To do that, we took advantage of the assay of our NAD level because we use a radio-labeled nicotinamide as a precursor to label the NAD. Therefore, it enabled us to do a post-trace assay to determine the fate or the half-life of the NAD after injuries. As you can see, the experience setup is like this. We first labeled axons for 3 days with radio-labeled nicotinamide. Just before the axon injuries, we wash away the radio-labeled nicotinamide, but then trace the axons with unlabeled nicotinamide. By doing so, we're actually measuring the half-life of nicotinamide in the axons compared to the injured and uninjured conditions. As you can see in the blue lines, in the uninjured conditions, the NAD is actually very, very stable inside of axons, with a half-life longer than 24 hours, as far as we can go here, as I should spot here. But in sharp contrast, after the axon injuries, there's a dramatic decrease of the half-life of NAD inside of axons, with the half-life of NAD shift to about 3 hours with the axon injuries. This suggesting that actually the injury, or the axon injury in particular, active the consumption of NAD, active process of consumption of NAD in the axons. And to further support this hypothesis, we can measure the fate of the radio-labeled NAD after the injury. Similarly, use our Purp3 assay. As you can see, after the injury, we can measure the level of all the NAD precursor together with the intermediate inside of the whole culture system. As you can see here, the black bar shows the NAD level inside of axons, while the white bar shows the nicotinamide level inside of axons. You can see after injury, well, there's a rapid drop of total NAD level as shown by black bars. There's actually a corresponding increase of nicotinamide inside of axons, suggesting or further suggesting that is after the axon injuries, the NAD got actively consumed and turning back into its precursor, nicotinamide. And with that information, we then make our focus onto the enzymes or the event that catalyze this breakdown or the consumption of NAD to its precursor nicotinamide. Basically, we are focusing on the NAD consuming enzymes. There are two major families of NAD, before I go into that, we also were able to show that this active consumption of NAD, similar to the NAD depletion event, is functioning downstream of thermal mechanism pathways. Because when we do the NAD measurement or the NAD half-life measurement, you can see in the control conditions, the NAD half-life always going to about 3 hours after the injury. While in the thermal knockout, the 2MAP2K knockout, or the 3MAP2K knockout, jump 3, you can see the half-life of NAD got significantly increased, bring to about 8 hours, something like that, after injuries, suggesting this activation consumption of NAD is indeed functioning downstream of thermal mechanism pathways. And then, as I just said, we make our focus to identify the event or the molecules or the proteins that cause the consumption of NAD inside of axons. To do that, we took a very simple but a little bit tedious approach. We got all the antibodies we can purchase through in the community or required from request from the community that recognize all the NAD consuming enzymes. That is two families. The first family is the Sirt2 family. The next family is the Parp family. Through this very simple but some little bit tedious approach, we were able to identify that is, first, all the 7 members of the Sirt2 family members are expressed in the neurons, but some of them show different localization. For example, the Sirt1, which is already expressed in the total neurons, but when we fraction the neurons or use this or very trick to isolate the axons from the neuronal cell bodies, you can see the Sirt1 actually is exclusively localized in the neuronal cell bodies, but not in the axons. And similarly, when we do the same thing for Sirt6, it's also the Sirt6 is exclusively localized in the cell body, but not axons. But at the same time, you can see our operation immediately, many of the Sirt2 family members and many of the Parp family members are indeed present in the axonal fractions. For example, the Sirt2, the Parp6, and the Parp11, or something like that. With that information, we then try to identify or explore the possibility that one or more of its family members of NAD-consuming enzymes may participate in the NAD consumption after injury in the axons. To that end, we perform a SHR screening or candidate SHR screening to identify the search of Parp family members that knockdown of them may delay the axonal degeneration after the injury. And we were lucky that is, we were able to identify 2 family members, the Sirt2 and Parp3, that when knocked down each of them, they actually cause a partial deletion. They cause a partial delay of axonal degeneration compared to the control conditions. But they seem to function. These 2 proteins seem to function additively because when we knock down both of them together, they further enhance the protection of the axonal, they further delay the degeneration of axons after injuries. This gave the first indication that Sirt2 and Parp3 are the two major NAD consuming enzymes that may involve or might involve in the axon degeneration after the injury. And supporting the idea that the Sirt2 Parp3 are indeed required for the active NAD consumption after injury, as well as for the ATP depletion after injuries. We got the Parp3 knockout mice, and on top of that, we use the shRNA to deplete the Sirt2. As you can see, when we measure the NAD half-life in injured axons compared to the control condition, there is also a similar shift or the significant increase of the half-life of NAD inside of the axons after injury when we deplete these two NAD consuming enzymes. And correspondingly, when we deplete these two NAD consuming enzymes, there is a corresponding increase or there's corresponding delay of the ATP depletion inside the axons compared to the control conditions, suggesting these two NAD consuming enzymes may be indeed required for these metabolic events going on in the damaged axons. And then finally, we're going to the in vivo assay, to the mouse model, to determine whether these two NAD consuming enzymes may indeed regulate the pathological axon degeneration of the axons in vivo. And we just got some preliminary results showing the answer is yes. Here, we first use the fluorescence fluorescence in situ to identify the expression profile of the Sirt2 and Parp3. Here shows example of the ganglia, the sympathetic ganglia. As you can see by the fluorescence in situ together with the co-staining of the TRK marker for the neuronal cells, you can see there's a perfect co-localization of the expression of Sirt2 and Parp3 with TRK labeled neurons, suggests they are indeed expressed in the neurons, at least in the sympathetic ganglia. And then in an in vivo assay, for the degeneration of the axons after the injury, we use a model that we can determine the degeneration of the sympathetic axons after the physical injuries. To that end, we can here show the diagram of the model system. As in the mouse brain, there is a superior cervical ganglia, which is a sympathetic ganglia, that have their axons, one of the branch of the axons projecting into the brain, which control the contraction of the blood vessels that providing the blood flow into the brain. And this projection is important to control the blood flow and to make sure the blood flow match the requirement or the demand for the energy inside of the brain. We can surgically remove the cell body localized in these superior cervical ganglia. By doing so, shear the axons from the neural cell bodies, and then ask what's the fate of these axons after its damage. As you can see, in this control condition, when we analyze this region of the blood vessels, which have all the mesh, heavy dense of mesh of sympathetic axons encasing the blood vessels, after injury, almost all the sympathetic axons will degenerate in 2 days. But in contrast, or in very sharp contrast, in the Sirt2 Parp3 double knockout mice, this degeneration process is largely delayed. It provides the first piece of evidence in vivo that the Sirt2 Parp3 indeed participate in the degeneration of axons, at least for the sympathetic axons in vivo. So taking all the story together, today I'm showing to you that is, in the first portion of my research in Dr. Lavan's lab, we were able to determine or delineate a signaling pathway that is novel for the degeneration of axons after pathological insult. And in my ongoing research, we are trying to make a connection or a metabolic connection between the degeneration event together with the NAD consumption inside of axons. And for the sake of time, I'm going to tell you is that we also have evidence suggesting the NAD production or NAD synthesis is also a key event inside of axons, because depletion of a key NAD synthetic enzyme, which called NMNE2 in the axons, can cause a spontaneous degeneration of axons without any damage, further suggesting that NAD is essential for the survival of axons under the normal physiological conditions. And with that, I would like to first thank you, my mentor or postdoc mentor, Dr. Lavan, for his support of my postdoc research at the Rockefeller University. And my ongoing research at Peking University is supported by the School of Life Sciences, the Joint Center of Life Sciences, the McGovern Institute, as well as the National Science Foundation. But most of all, I always feel privileged to work with some of my very talented graduate students, which have been forming the core and the foundation of my research group. So thank you for your attention.